Hello everyone. Today we are going to talk about the anatomy of the prokaryotic cells. We are specifically going to concentrate on bacteria. Most bacteria aren't dangerous for humans. Many of them live on or inside of our body, and they actually help us to stay healthy. For instance, there are bacteria that can help us digest food in our gut. And some of them even produce vitamins like vitamin B and vitamin K. And vitamin K is actually known to help us clot our blood. Some bacteria are even used to produce food such as cheese and sauerkraut, yogurt, pickles. Some of them produce things like ethanol and acetone and even oxygen. Actually, less than 1% of all the bacteria out there are responsible for the diseases, and we refer to them as pathogenic. So the term pathogenic means disease causing. And again, not only bacteria, some bacteria out there are known to be pathogenic. We also have viruses. We are in midst of a pandemic right now, COVID-19 virus. So viruses are also pathogenic. And so pathogenic just means the ones that would cause harm to us, disease. Some other benefits of those microorganisms is we can actually use some of them to produce things that would help us humans, for example, insulin. Now, this idea of using a bacteria to actually get a human product such as insulin is due to the fact of a technology known as recombinant DNA. And that's a technology scientists developed that made it possible to actually insert a human gene into the genetic material of a bacterium. And so we refer to that as recombinant microorganism. And that recombinant microorganism would be able to produce the protein encoded by the human gene that was inserted in it. And that's actually what they did with the insulin production to accommodate the high number of diabetics that are in need of insulin. They took the insulin human gene, they inserted it into the bacteria, and the bacteria started making the human insulin. So we see how microorganisms can actually be also used in the COVID-19 vaccine. For one of the vaccines that are now, got developed, they actually went ahead and they took a piece of the COVID virus, they inserted it into an adenovirus, which is a common cold virus. And so when they inject it eventually into the host, when they inject that adenovirus into the host is going to start making antibodies against the COVID-19 virus. So as you can see, having knowledge of these microorganisms can actually benefit us. We can take advantage, as I said, of inserting genes from different organisms into bacteria or into viruses so that we can start making products like insulin, or we can start making antibodies. So it's definitely helpful for the pharmaceutical companies, for the health industry, even for the food industry as well. Like if we have knowledge about those microorganisms, we're going to be better off in preventing food spoilage. We're also going to talk about biofilms in this class. So biofilms, that's when microbes attach themselves to a surface and they grow in masses. And basically, biofilms, you can find them everywhere, even on your teeth. So when you guys floss and you find that white stuff that's coming out, that's basically bacteria. That's bacteria that was stuck on your tooth. And if they actually get stuck on your tooth for a while, let's say due to you not flossing a whole lot, you have to go to the dentist and they have to scrape it off, right? So when they scrape it off, that's basically biofilm that they are scraping off. You can even find biofilm, like a bunch of bacteria growing even underwater. And biofilms can actually cause infections because you end up with a community of microbes. So a biofilm, you don't just have one type of bacteria, you can actually have multiple types of bacteria, and they kind of create that strong community. So it's really hard to actually get rid of a biofilm, and they can cause pretty bad infections, and they can be resistant to antibiotics. And that's an image right here of biofilm on catheters. Actually, catheters are like the worst things ever. I mean, they are pretty important, but they do cause lots of infections to patients. Almost all patients having a catheter would get an infection, and it's just because that catheter is placed in there for for a period of time, and it's usually like a long period of time, and so the bacteria starts to grow within the catheter, and eventually it can creep up into your body and therefore cause an infection. Now, the microbiota, we refer basically to the microbes that live inside of us or on our skin as microbiota or microbiome. And this is an image right here of the microbiota that you would actually find on the surface of your tongue. So basically what you're seeing, all of these are different types of bacteria that you could actually find on your tongue. And so actually this microbiota that you are seeing right here actually help us maintain good health. Without it, we could actually start falling ill. But the next time you share a glass of water with someone, you're not just sharing water, you're also sharing your microbiota with them. Let us take a look at some of the shapes and different arrangements for our bacterial cells. Some bacterial cells actually look like stars, and some of them look like rectangles. Some bacteria look spirally and they're pretty thin, so we call them spirochetes. If it's spirally but it's a bit thicker, we call it spirillum. Vibrio, those look like curved rods. When a bacteria looks spherical, we call it coccus. Coccus is singular. Cocci would be plural. If there are two attached to each other, we refer to it as diplococci. If it's a chain of spherical bacteria, we refer to it as strepto, so it would be streptococci. If it's a group of 4, we refer to it as tetrads. If it's a group of 8 to 32, we call it sarcinae. And if it's basically a cluster of spherical bacteria, cluster, we would refer to it as Staphylo, so Staphylococci. Bacillus, those are bacteria that look like rods. If you actually have 2 rod bacteria next to one another, attached to one another, we would call them diplo, so diplobacilli. So bacillus is singular. Remove the US and put an I, it would be bacilli, and that would be plural. So If you have, as I said, two of them attached to each other, you would call them diplobacilli. If you have a chain of bacilli bacteria, you would call it streptobacilli. Some of them actually are known as coccobacillus because they kind of look like they're not totally rods, and they kind of look like coccus bacteria, but they're a bit elongated, so we call them coccobacillus. And so, as I said, if you have two, call it diplo. If you have a chain, it's strepto. If we take a look at the structure of a prokaryotic cell, all prokaryotic cells, they have plasma membrane. They have cytoplasm, which is the fluid inside of the cell. They have ribosomes, and the ribosomes of prokaryotes, we refer to it as 70S ribosomes. Humans and eukaryotes have ribosomes, but we don't call it 70S. It's 80S for eukaryotes. All prokaryotes have DNA. Now, of course, the DNA of a prokaryote is going to look different compared to the DNA of a eukaryote. Most prokaryotes have plasmids, and plasmids are basically circular DNA. It's not attached to the original DNA, and those plasmids usually carry antibiotic resistance genes. So it's pretty important, and actually, not all bacteria have it though. And some bacteria can actually exchange their plasmids, and we will talk about it. Some bacteria have fimbriae, which are found surrounding the cell, like so. We're going to talk about their functions in just a few. Some of them have those projections known as pilus, or plural would be pili. Some of them have flagella, which is going to help them move, but there are other means of movement, and not all of them have flagella. A cell wall is going to be on top of the plasma membrane, and on top of the cell wall there is a layer known as the capsule. But again, not all bacteria have the capsule layer. Having the capsule layer is usually going to protect the prokaryotic cell even more against the immune system. And so we always say that the capsule is going to add to the virulence of the bacteria, and it's going to definitely play a role in making the bacteria more pathogenic, meaning the bacteria can cause disease if it has a capsule. Bacteria can produce and secrete something known as a glycocalyx, which is a sugar coat, and it would surround the bacteria. It would be external to the cell wall of the bacteria. The glycocalyx could be composed of polysaccharides, just basically sugars. It could also be made out of polypeptides, which are proteins, or a glycocalyx could actually be composed of both polysaccharides and polypeptides. Now, if that glycocalyx is firmly attached to the cell wall, we call it a capsule. And if it's unorganized and loosely attached, we refer to it as a slime layer. We can actually detect the presence of a capsule by doing a negative stain. So a negative stain would allow us to be able to spot a capsule. Now, if bacteria do have capsules, which, as I said, they're going to be attached to the cell wall of the bacteria, those capsules, they usually protect pathogenic bacteria from being phagocytized. What is phagocytosis? Phagocytosis is the process of let's say the immune cells engulfing something foreign or engulfing a bacteria cell. So when bacteria cells have capsules, that actually helps protect them from those phagocytic immune cells that we have in our body, or within the host, basically. So therefore, having a capsule definitely contributes to the virulence of the bacteria. And a lot of the bacteria that are actually capsulated, like they have a capsule, they can cause diseases versus bacteria that are not carrying a capsule. Because, as I said, the capsule protects the bacteria from being engulfed by the immune system of the host. It also protects it. It gives it extra protection from harsh environmental factors, so it's able to survive longer. So an example, so as I said, capsules can prevent phagocytosis, which is again the immune system of the host usually has what we call phagocytic cells, and those phagocytic cells, they engulf bacteria, they engulf toxins, they can basically engulf anything that kind of looks foreign. So having a capsule can prevent the bacteria from actually being engulfed by the immune cells of the host. Having also that glycocalyx can help form a biofilm, which is basically the bacteria attaching to surfaces. Bacteria can attach to so many different surfaces, whether it's the plants, rocks, the teeth, skin. So bacteria are found everywhere, and they can attach to so many different surfaces. Having a glycocalyx can definitely protect the bacteria from getting dehydrated and from losing nutrients. So it gives it extra. The bacteria Streptococcus pneumonia could either carry a capsule, or some of them don't carry a capsule. The ones that are encapsulated, those are the ones that are going to cause pneumonia, because the capsule is going to be protecting the bacteria from phagocytosis. The ones that are not carrying a capsule, those are usually going to be phagocytized pretty easily by the host, and so therefore they're not going to cause pneumonia. Some bacteria have flagella. Flagella are appendages that are attached to the external part of the bacteria, and those flagella, they propel and they help the bacteria basically move and swim. Flagella, they're made out of a protein known as flagellin. Not all bacteria have flagella. Some bacteria move by other means that we are going to talk about. Now, some bacteria have what's known as Fimbriae, and those Fimbriae, as you can see, they would surround the surface of the bacteria. Those Fimbriae, they're hair-like appendages, and they allow for attachment. They basically have a tendency to adhere they would allow the bacteria to adhere to other bacteria, or they would allow the bacteria to adhere to surfaces. And so having Fimbriae definitely makes the bacteria involved in forming biofilms, and that's when bacteria stick to surfaces, kind of like your teeth, rocks, you know, so many different surfaces bacteria can attach to. And having those Fimbriae, again, can help the bacteria basically attach to all of these different surfaces. They can even allow the bacteria to even attach to the lining of your small intestine. So E. coli, actually has those fimbriae around its surface, and therefore E. coli can actually stick to the lining of your small intestine and cause diarrhea. When those fimbriae are absent, usually no disease would occur because you wouldn't have colonization, because having those fimbriae will definitely help the bacteria colonize pretty quickly and grow pretty quickly on the surface. And definitely if they are colonizing your insides, that can cause problems. Some bacteria have what's known as pili, and those pili, they're a bit longer compared to the fimbriae, and those pili, they are involved in motility, so like they can help the bacteria move. Another thing is that those pili can actually help the bacteria transfer its own DNA to another bacteria. So let's say here are two bacteria right there. Notice how they're both sticking out their pili, and what happens is that their pili could actually connect, and it kind of creates a bridge. Notice how they are creating a bridge between them. And what's really interesting and cool about that is that this bacteria, let's say, is carrying an antibiotic resistant gene that this bacteria is not carrying. What's going to happen is this bacteria can actually take its antibiotic resistant gene and it can actually transfer it over to this bacteria right there. And if this bacteria is carrying, again, a gene that, let's say, makes it resistant to heat, for example, it can also pass it on to the other bacteria. And so what happens is when bacteria do that and they start exchanging DNA amongst one another, if we are trying to, let's say, get rid of those bacteria, it's going to be a very hard task for us because those bacteria are going to be changing on us very quickly since they are exchanging their DNA and basically getting new characteristics and new properties that they didn't have before. So it makes it, again, a harder job for us to get rid of those pathogenic bacteria because, as I said, they could actually start exchanging antibiotic resistant genes. Now, if we talk about the structure of the cell wall of the bacteria, there are differences between the cell walls of different bacteria, and we can actually figure out those differences and figure out the type of cell wall that the bacteria is carrying by doing certain stains, like a Gram stain can tell us some differences between the cell walls of the bacteria. Having a cell wall actually contributes to the virulence of the bacteria, and we're going to talk about how. The cell wall of a bacteria is made up of something known as peptidoglycan. Peptidoglycan is basically consists of disaccharides, which are basically sugars, and polypeptides, which are proteins. Having a cell wall can actually prevent the bacteria from osmotic lysis, which basically means it can prevent the bacteria from intaking too much water, so it can prevent the bacteria from basically swelling up and bursting. So those are the main components of the cell wall. As I said, the cell wall, it's made up of disaccharides. So those rods that you're seeing right here, those are the disaccharides. Those are basically the sugar portion. And then those blue kind of greenish beads that you are seeing, those are representing the polypeptides. Those are the proteins. Those are the sugars that the peptidoglycan is made out of, and again, they're represented by those rods right here, and then those are the polypeptides. Now, when we look at a gram-positive cell wall, The gram-positive cell wall has many layers of that peptidoglycan versus the gram-negative. They only have a very thin layer of that peptidoglycan. So right here, if this is a gram-positive bacteria, it would have, first of all, a plasma membrane. On top of the plasma membrane, you would find the cell wall. So as you can see, the cell wall of a gram-positive, it's definitely made up of several layers of those disaccharides and the polypeptides. So you can see it's pretty thick. Gram-positives also has gram-positive bacteria also has those acids right here known as teichoic acids and lipoteichoic acids. Those teichoic acids, they basically provide they act as an antigen, meaning that they're pretty specific. So each bacteria could have slightly different teichoic acids. And so therefore they act as a self-antigen for the bacteria. And basically that antigen is going to note that this bacteria is kind of different from the other bacteria, depending on which exactly type of teichoic acid is it carrying. And so it could actually make it possible for someone to identify exactly which gram-positive bacteria based on the teichoic acid that it's carrying. Because again, as I said, it can work or it's going to act as a self-antigen for that bacteria. Biggest difference between a gram-positive and a gram-negative is definitely that cell wall structure. So first of all, there would be a plasma membrane, and then notice how the gram-negative only has one layer of that peptidoglycan. So that's why we say gram-negative. They have a very thin peptidoglycan. But what else do they have? On top of the peptidoglycan, they have what's known as an outer membrane. And this outer membrane consists of some polysaccharides, and it consists of lipids, and we call that lipid A. Now, having that outer membrane within the gram-negative can actually help the gram-negative bacteria evade phagocytosis. Having that outer membrane can actually help the gram-negative bacteria create kind of like a barrier. So that outer membrane kind of acts as a barrier to antibiotics, for example, penicillin. It acts as a barrier against digestive enzymes. And so you can see that when gram-negative bacteria have that outer membrane, you can see how much that outer membrane is basically going to be helping out that gram-negative bacteria evade the immune system of the host, evade basically getting broken down by enzymes that the host again might throw at that bacteria right there. Another thing that's going to, help out the bacteria is that lipid. Lipid A is basically that lipid portion of that lipopolysaccharide, and that lipid, as you can see, it is embedded in the outer membrane of the gram-negative bacteria. And so what happens is when the bacteria, let's say a gram-negative bacteria dies, when that bacteria dies, they actually release their lipid A, which is going to act as an endotoxin. And so you can see that even if the host succeeds in killing a gram-negative bacteria, that gram-negative bacteria, if it's killed, it's actually going to release that lipid A, and that lipid A is going to, it's basically a toxin to the host. And so that lipid A is definitely responsible for a lot of the symptoms associated with getting infected by a gram-negative. Because again, as the host immune cells are trying to protect the host, and they're going to do their best trying to kill that gram-negative bacteria, If some of those gram-negative bacteria, they start dying, they're going to release that toxin, which is the lipid A, and some of the symptoms that you would, for example, have is fever, shock, dilation of your blood vessels, blood clotting. So you can see it's a huge list of negative side effects that the individual would face if that lipid A, again, is released. Another thing attached to that lipid A, there's this O polysaccharide, and that O polysaccharide basically acts as a self-antigen. So it kind of acts as an antigen that's going to help us identify this bacteria. So it would help us distinguish which gram-negative bacteria we are talking about. And so that O polysaccharide is kind of acting the same as the teichoic acid in gram-positive bacteria. So the teichoic acid in gram-positive and the O polysaccharide in the gram-negative, they kind of have the same function, which is, again, they would help us identify which type of bacteria we are talking about. Now, bacteria, they do have DNA, but other than having that circular thread of DNA, they also usually have what's known as plasmids. And those plasmids, they are basically extra chromosomal DNA, meaning that they are not part of the original DNA. And those plasmids, they usually carry genes for things like antibiotic resistance, tolerance to toxins. Those plasmids can even be transferred from one bacteria to another, again, via those pili that we've talked about previously. And so therefore, those plasmids are pretty important, and they are definitely beneficial if a bacteria carries those plasmids. Now, when essential nutrients are basically getting depleted from a bacteria, the bacteria is going to go ahead and start producing what's known as an endospore. Endospores are usually produced by gram-positive bacteria, such as the ones under the genre or genus Bacillus and Clostridium. So those are usually the ones that produce those endospores. Endospores, they are basically more durable, kind of daughter cells, if you can think of it this way. And those endospores, they have very thick walls that make them basically able to tolerate high heat. They're able to tolerate like harsh environmental conditions. Those endospores, they're made internally within the bacteria cell membrane, and they can actually get released into the environment. And once they are released into the environment, they can actually survive on their own. And as I said, they can survive extreme heat, they can survive lack of water, they can actually tolerate a bunch of toxins and radiation. So as you can see, getting rid of the endospores is not an easy task. They've actually found endospores that were 40 million years old, and when they took that endospore back to the lab and they've given it the nutrition that it needs, it was able actually to germinate back. So let's talk about the process of forming those endospores, and that process is known as sporulation. But first, let me kind of show you an image. So this is a bacteria forming the endospore inside of it. So one more time, the endospore is going to be developed inside of the bacteria. Next, let's discuss Archaea. So Archaea, they're also considered prokaryotes. They do have cell walls, but their cell walls don't have peptidoglycan. They're very similar to bacteria, but Archaea, they usually live in very harsh conditions. So they love to live in such extreme conditions. For example, they love to either live in a very salty environment, and so they thrive in high salt concentrations, and so we refer to them as halophiles. Some of them, they love to live in like super hot conditions, like near a volcano or hot springs where it's extremely hot, and so we refer to them as thermophiles. And some of them love to live in very cold conditions. Some of them we refer to them as methanogens, and those are the ones that usually produce methane gas, which is a gas that has a pretty foul odor to it. Not a lot of, actually, I should say we didn't till now find any archaea that are pathogenic, disease-causing archaea, and that's why we don't give too much attention to them because they don't really harm us. So most of the time you're going to find that scientists are more concerned about learning about microorganisms that either harm us, so we can understand them better, or, if they are going to benefit us in a way. So that's why we're not going to talk too much about Archaea in this class.